Hi, everyone. I'm Andy Jones. I'm a PhD student at Princeton working with Barbara Engelhardt. And today I'll tell you about a computational framework we've developed for aligning spatial genomics samples as well as histology data. And this is joint work with Will Towns and Didong Li, who are postdocs with Barbara. So to give you a bit of background on spatial sequencing technologies, although Barbara gave you some at the beginning, I like to use this analogy between sequencing technologies and fruit-based dishes, which some of you may have seen before. But bulk sequencing technologies on the left here profile expression levels of an entire tissue. So this will yield a gene expression or a protein expression vector across the entire tissue. And this is sort of analogous to a smoothie where all the fruit have been blended up together. Single-cell sequencing in the middle here has also become popular for characterizing the phenotype of individual cells. And so this is sort of analogous to a fruit dish, where now each piece of fruit is distinct, but still they're sort of bundled together. And most recently, spatially resolved sequencing has become popular, which I'll talk about today, which allows us to profile the phenotype of individual cells as well as collect their spatial location within the tissue. And so we can think of this as analogous to a fruit tart, where each piece of fruit is distinct, and also we know the spatial organization of each piece. And so although spatial sequencing technology is still somewhat in its infancy, it holds quite a bit of promise for being able to understand things like cell-cell communication, how structural patterns in a tissue relate to functional patterns, and how these things change across individuals, diseases, treatments, other types of perturbations. So these are just three recent examples from three recent papers where spatial gene expression profiling was used to localize different cell types, characterize local response to different types of perturbation, as well as study developmental trajectories spatially. And so despite all these possible applications of spatial sequencing, there are actually still very few computational techniques to analyze these type of data. And the techniques that do exist tend to focus on analyzing just one sample, where here a sample refers to a single tissue slice. But in order to leverage all of the information in these data, it would be useful to be able to jointly analyze spatial sequencing data across multiple tissue slices, and possibly even from multiple donors and things like that. So on the left here, we can see that when the spatial coordinates of multiple samples aren't in the same coordinate system, we have to analyze each slice separately. But on the right, if we were able to align these samples within the same coordinate system, we could then analyze these jointly. And to motivate why aligning samples in the first place is a good idea, here are three possible new types of analyses that are possible after alignment. So first, the most obvious one is that by aligning multiple samples, we gain increased statistical power, which is useful broadly for different types of statistical techniques you might use on these type of data. Second, once we have samples aligned within a common coordinate system, we can get an estimate of the variance at a particular location in space, as well as across dimensions in space. In particular, perhaps across the z-axis of space as opposed to just the x and y axis. And third, as we collect more and more of these spatial genomic samples, it creates an opportunity to create spatially resolved atlases, where here we can think about an atlas being as a queryable database where we can query for any spatial location and receive the phenotype of the tissue at that location, where the phenotype could be a number of things like gene expression levels, protein expression levels, certain morphology characteristics, and things like that. So as I mentioned, without any modification, it's usually unlikely that multiple tissue slices will reside in the same coordinate system. And there are multiple reasons for why this spatial distortion can pop up. And we generally can think of this as two different sources of distortion. The first one being technical distortion. Often this is due to tissue preparation, tissue handling, or even during the sequencing process. And the second one is biological variance, or biological distortion, which is inherent to the tissue itself. And roughly, we can sort of, in theory, decompose this further into interesting and uninteresting biological variance, where in an alignment, we would ideally remove the uninteresting biological variance. And so there's a need for methods that will align these samples to allow for these downstream analyses. So to address this problem, we developed a model which we call Gaussian Process Spatial Alignment, or GPSA. And our model basically consists of two primary components. The first component on the left here is a model that we call the warp model. So in this part of the model, each tissue sample is given its own warping function that warps its own spatial coordinates into a common coordinate system in the middle in yellow there. And the second part of the model captures the phenotype of this tissue within that common coordinate system at each location in space. So here we model the spatial organization of this phenotype with a single function, f, across all of the samples, which captures this shared phenotypic structure across all of the samples. We place Gaussian process priors on each of these functions for both the warping functions, G1 through GS, as well as the phenotype function, f. And so this results in this two-layer deep Gaussian process where in the middle layer we capture this common coordinate system with aligned spatial coordinates. To give a visual demonstration of how our model works in a simulation study, we applied it to this synthetic data set with two simulated tissue slices where the two samples originally came from a grid of spatial locations but had distorted spatial coordinates. And so in these diagrams here, each point corresponds to a spatial coordinate, and it's colored by a synthetic one-dimensional phenotype here. And in the animation, we can see that our model, GPSA, is aligning the spatial coordinates of certain points with similar phenotypes at those points. Our model also allows for two different types of alignments. The first type of alignment, which we call a de novo alignment, learns the warping functions completely from scratch without any prior knowledge about the common coordinate system, although we can initialize this with a reference sample if we like. And so this can be useful when an established common coordinate system doesn't exist for a particular tissue type or organism. And the second type of alignment, which we call the template-based alignment, uses a known template coordinate system to map these distorted spatial coordinates into that given common coordinate system. Moving on to spatial transcriptomics data, we applied our model to a series of data sets, so I'll walk you through those results now. We first applied it to this data set from Ståhl et al. In 2016, which profiled a series of breast cancer biopsies using spatial transcriptomics. And so we first applied synthetic warps to their spatial coordinates and used the phenotypes given by the data, and we checked whether GPSA could correct for these warps. And we found that on the right, GPSA could correct for these very local and nonlinear distortions that occur in the spatial coordinates. And we also found that our model could better correct for these distortions compared to a competing method called PASTE, which uses a purely linear alignment. Looking across different types of warps, so there are many different types of distortions that might appear in real data. We checked how well GPSA could align different types of warps, and we found that across three different types of spatial warps, we could pretty reliably recover a good alignment. And so we sort of hypothesized that this flexibility across multiple different types of warps and having this sort of local nature will be important. So then we fit GPSA to align all four slices from this breast cancer data set. Again, this is spatial transcriptomics. We fit our model, we extracted the common coordinate system, and then we looked at this common coordinate system to see what sort of new analysis we could do. And so one of the first things we did was look at the variance of gene expression across space along this z-axis for all genes. And again, this is the type of analysis that is only possible after aligning these slices into a common coordinate system. And we found substantial variability across this axis for many genes, including, there we go. So yeah, we looked across all genes, and we saw exceptionally high variability for a small subset, and these tended to correspond to gene sets and pathways that are known to be associated with breast cancer. Having characterized the spatial variants in these breast cancer slices, we next asked whether we could actually use our model to now create a three-dimensional model of the spatial coordinate system. So to do this, we basically use the probabilistic nature of our model to densely impute gene expression within this common coordinate system. And so this is basically possible due to our warping model as well as our phenotype model that captures this spatially resolved phenotype within all of the slices. And so imputation here is straightforward with our model, and on the right here we see a dense imputation for one gene, FN1, where each point is colored by the estimated expression at that point. And so we see this direction of creating 3D atlases from 2D slices as an important direction in the future. We next applied our model to another data set from the 10x Visium platform that profiled two slices of the mouse cortex. So we, again, fit our model to these data, extracted the common coordinate system, and looked at sort of what changed between the original spatial coordinates and the aligned spatial coordinates. And we found that our model corrected for very local distortions that occurred in the cerebellum folds in these brains, and that these were sort of highly nonlinear and local distortions was important to be able to capture across this tissue. With the same data set from the mouse cortex using the Visium platform, we also asked whether we could use our aligned spatial coordinate system to improve our downstream analysis of these type of data. So as one example of this, we looked at our ability to identify spatially autocorrelated genes before and after alignment. So here we use Moran's I statistic, which is a measure of spatial autocorrelation for each gene. And we saw a near universal increase of the spatial autocorrelation scores after alignment, and even some of the gene autocorrelations became statistically significant after alignment. As one of the last applications I'll talk about, we attempted to apply our model to very high resolution data. So this is from the SlideSeq V2 technology from Stickels et al. In 2021. This is from the mouse cerebellum, again with, this is measuring spatial gene expression. And again, we found our model corrected for major distortions that existed in this data, most notably in the dentate gyrus. So on the top row here is the unaligned samples, on the bottom row is the aligned samples, colored by specific genes. And so again, we're correcting for very local and nonlinear distortions in this mouse hippocampal data. To look at this alignment in the hippocampus more quantitatively, we computed the distance for specific landmarks before and after alignment. So here we used certain corners of the dentate gyrus as well as the area CA1 and looked at the distance between these landmarks, between slices before and after alignment. And all three of these landmarks became much more close to each other after alignment, which is sort of a nice validation that we're aligning known structures within these tissues. So our model also easily extends to account for multiple modalities of data if they exist. So what I talked about before is just spatial gene expression, but often you have multiple different types of phenotypic readouts across a certain tissue. So as one example of this, we also applied our model to the Visium data again, but this time we not only used the spatial gene expression data, we also included the H&E stains. And so we found that our model could align both of these modalities within the same common coordinate system, again finding the same sort of local alignment within the cerebellum. And so we envision this multimodality feature of GPSA to be important for understanding multiple different types of phenotypes within a common coordinate system. And as future work, we're now considering the question of experimental design under our alignment framework. So specifically, given that we can align sequencing readouts now, spatially resolved sequencing readouts, we're now asking whether we can optimize the data collection process itself. And in particular, we'd like to be able to identify the experiments we should run in terms of slice location and the data modality we should collect that could both maximize the fidelity of the reconstruction that we can make of the tissue, but also minimize the costs and the number of slices that need to be profiled. So bringing the talk to a close here, in this work we propose GPSA, Gaussian Process Spatial Alignment, which is a method for aligning spatial genomics samples as well as histology data and other modalities into a common coordinate system. It allows for flexible, local, nonlinear, and probabilistic warping functions. And with this alignment method, which can be de novo or template-based, we can construct 3D atlases from 2D slices with or without a reference sample. We can integrate spatial profiling modalities, and we can also look for axes of variation for specific markers. I'm participating in the comment-a-thon, so feel free to engage with the preprint on bioRxiv. And I'd like to thank all my collaborators. So this is joint work with Will Towns and Didong Lee. I'd like to thank our experimental neuroscience collaborators, Kate Pena and Addie Minerva, as well as my advisor, Barbara Engelhardt. You can find our preprint on bioRxiv, and our group is actually moving to Stanford and Gladstone this summer and is actively recruiting PhD students and postdocs. So contact Barbara or come find her here in the audience if you're interested. Thank you.
First question.
Hi, Andy. This is Jean Fan from Johns Hopkins. Really beautiful talk. I really loved your motivations. And yeah, I just wanted to ask if you could elaborate a bit more on the 3D imputation and, to what extent do you think, how far spaced do you think your slices could be in the Z domain and still kind of accurately reconstruct that, Z-level spatial variation?
Yeah, it's a great question. Yeah, we were somewhat limited by the available data to even answer the question of how far apart can slices be to be able to reliably impute. This is sort of something we're thinking about with the experimental design question as well, sort of how can you maximize the distance between slices while also accurately imputing between them. Our current hypothesis is basically that it varies a lot across space depending on how spatially variable a certain phenotype is across space. So if it's relatively constant across a part of space, you can get away with slices farther apart. But if it's changing rapidly in a region of space, you may need to take multiple samples in that region.
Thanks for a great talk. I had a question about your alignment of H&E and spatial transcriptomics. It's not super clear to me why you need that, because if you have like spatial locations for the spatial transcriptomics, right? So is it to sort of improve the alignment or to find like match features between images? And yeah, I wasn't super sure of what the purpose of that would be. Yeah,
actually those are kind of the two motivations. So one is to improve the alignment itself, which is more of a statistical motivation, I guess. So you can sort of think about it as increasing the sample size to help the alignment. So sort of more phenotypic variation in the data will help the alignment. The other reason is, if you're interested in looking at the morphology characteristics in the H&E stains, you can then easily map between these two parallel modalities. So those are sort of the two motivations, statistical and interpretation.
Very nice. Have you thought about how you could extend this to biological replicates? So from borrowing information across individuals, obviously it's not going to be possible or maybe advisable to map everything onto a one coordinate framework there, but one could certainly think about ways that you could borrow information sensibly so that the results across individuals are comparable. Have you thought about that? And is this something that you're thinking about in the future?
Yeah, it's a great question. I mean, there are a few things we've thought about. One, sort of human-in-the-loop way to do it is to think about annotating landmarks across individuals, which allows for some amount of coherence across alignments and individuals. If the tissue itself has a very canonical structure, like in the brain, often it's much easier to sort of get a very basic reference sample, but for more heterogeneous tissues, you may need sort of more well-defined landmarks to get that consistency across donors.
had the same question.
All right, so following up on the multiple sample question, I think it's possible to consider like a morphological differences across individual. Like for example, if you have multiple slices from disease individual versus multiple slices from like a healthy individual, try to understand morphological changes and like some expression changes associated with that kind of change is going to be somewhat like insightful, I think. And have you ever looking at that kind of stuff, or like is the model like flexible enough to quantify such changes across individuals?
Yeah, it's a good question. We haven't modeled perturbations like that explicitly in this model. Right now, we sort of assume that you have, only healthy or only disease that you're aligning, but that's a natural next question. And the benefit of our model is that it is flexible enough, I think, to be able to accommodate some model for perturbation of disease, injury, drug, whatever. So that's a natural next step, yeah.
Hi, great talk. This is Leonardo from Liber Institute for Brain Development. I'm just curious about if you're considering using images from multiple technologies like SMFish plus H&E, and then also if you're cautious about the fact that sometimes your slices the tissue can fold. So for your future experiments, if you have a critical missing piece in the middle, how that would affect.
Yeah, it's a great question. Yeah, there are all types of distortions that can happen. We don't have an explicit model for that type of folding. It's possible that the model could capture that in a data-driven way right now. Or if you have like a missing piece of section in the interior of this tissue. To reuse the landmark answer, if you know that kind of distortion exists in your tissue, you could go in and explicitly fix certain parts of the common coordinate system to account for that. Or you could check whether it can learn it in a data-driven way.
Well, actually, that was already addressed by the previous question. Thank you.
Okay, thank you.
Thanks.